Lecture 8 is going to cover the cardiovascular system. This is going to go over two chapters, chapters 18 and 19, but it's only going to be certain sections from those chapters. So we're going to start with chapter 18. This is the part of the cardiovascular system involving the heart. So the heart is part of two circuits, the systemic circuit and the pulmonary circuit. The systemic circuit is the circuit that pumps blood from the heart to the rest of the body. So going from the left ventricle, going up through the aortic arch and then down the descending aorta, and then making its way down to the rest of the body, where it then branches, those arteries branch off into capillaries, and those are the ones that feed the organs and tissues. The pulmonary circuit is the part of the circulation that takes blood from the heart to the lungs where it gets oxygenated and then comes back oxygenated back to the heart. As far as the blood flow, you should be familiar with the blood flow throughout the heart. Okay. And there's really no particular order that you should start at, but usually I like to start here at the right atrium. That's where the blood drains from the superior vena cava and the inferior vena cava. The inferior vena cava is the one that brings the blood back from the abdomen and the limbs. The superior vena cava brings the blood back from the neck and the head, but they both drain here at the right atrium. From the right atrium, it goes through this valve. It's called the tricuspid valve, or also known as the right AV valve. From there, it goes into the right ventricle. Then the ventricle contracts, pushes the blood through this pulmonary semilunar valve. It'll go through the pulmonary trunk, and then it bifurcates into two pulmonary arteries. From the pulmonary arteries, it's going to go into the lungs, and that's where the gas exchange is going to occur. So it'll lose the carbon dioxide, will exchange the carbon dioxide for the oxygen. Then once it's oxygenated, the blood goes back into the heart via these pulmonary veins. So here it goes from the veins, it goes into the left atrium. From the atrium, it's going to go down, so it's going to go into the left ventricle, and it passes through this valve that's called the bicuspid valve or the mitral valve, or you can also refer to it as the left AV valve, AV for atrioventricular. So it's also described that way. From the ventricle, it's going to push the blood up through the aortic semilunar valve, and then it's going to find its way through the aorta. So once it passes the aortic arch, it flows down through the descending aorta. One of the distinguishing characteristics of cardiac tissue are the intercalated discs. So these are structures that contain junctions between cardiac cells. So they could contain desmosomes, which help hold the cells together. So it prevents them from separating during contractions. And then there's the gap junctions that allows the cells to communicate with one another. So this allows the heart to be a functional syncytium. A functional syncytium is a group of cells that are able to communicate with one another and perform coordinated tasks. So in this case would be like a coordinated contractions. Here you can see an example of cardiac tissue. You can see those intercalated discs that they have. That's one of the ways you can identify cardiac tissue versus skeletal tissue, because they're both striated, but the cardiac tissue is the one that has these intercalated discs. It also, the cells or the the fibers are also branched compared to skeletal muscle. Okay. But there's a lot of similarities between cardiac tissue and skeletal tissue. So as you can see, there's also the T tubules are here too in the cardiac tissue. The T tubules, which are the transverse tubules that we looked at last time. There's a sarcoplasmic reticulum is also here that stores the calcium. Then there's the different parts to the muscle fiber, like the Z discs, the I band, the A band. So those are all the same as the skeletal muscle fibers. Action potentials are also similar in cardiac tissue as they are in skeletal tissue, but there are some differences. So for example, the resting membrane potential here is not going to be flat like it was in the skeletal tissue. And also there's a different threshold that has to be reached. In skeletal muscle tissue, it was a negative 55 millivolts. Here it's a negative 40 millivolts. And the cardiac action potentials begin because of the pacemaker cells. So those generate a pacemaker potential, and that's just a slow depolarization that's due to sodium coming in and also the potassium channels, I mean the potassium going out. Once it reaches the threshold, then depolarization occurs, and that's when the calcium influx occurs. Okay, so those once it reaches the threshold, those calcium ion channels are going to be activated or opened, and then there's going to be an influx of calcium, and that's going to create the action potential. Repolarization is going to be due to the calcium channels inactivating, and then the potassium channels opening. So this means that the potassium ions are going to leave the cell, and so it's going to repolarize. It's going to go back to being negative. And then in order for a heart contraction to occur, there needs to be a signal that's generated at the right atrium. So in the right atrium, there's a group of cells that are known as pacemaker cells. These cells are the ones that generate the impulse spontaneously. And so that impulse goes from the sinoatrial node. So again, node is just a cluster of cells, and it travels throughout the atria. Then that impulse is going to reach the atrioventricular node. And then from the node, it goes into the atrioventricular bundle, which is also known as the bundle of His. This is the only electrical connection between the atria and the ventricles. So from that bundle, it'll branch off into the bundle branches, and then it'll conduct the impulse through the interventricular septum, which is this structure here that separates the two ventricles. So that's why it's called interventricular septum. From there, the branching continues, and then it goes into the subendocardial conducting network, and this is what depolarizes the contractile cells of the ventricles. So whenever there's depolarization of either the atria or the ventricles, that's going to precede the actual contraction of the chambers. So once the atria depolarize, then the atria will contract. And then once the ventricles depolarize, then they'll contract. Defects in intrinsic conduction could cause things like arrhythmias, which are irregular heart rhythms, which are due to uncoordinated atrial and ventricular contractions. It can also cause fibrillation, which is rapid, irregular contractions. And this is when the heart becomes useless for pumping blood and it causes poor circulation, or it'll stop circulation. This could be treated with defibrillation, which interrupts the chaotic twitching and it gives the heart a clean slate to start a regular normal depolarizations. The electrocardiograph can detect electrical currents that are generated by the heart. So there's an electrocardiogram, which is the ECG or EKG, and this is the graphic recording of electrical activity. So this is the activity you guys did in lab. But this is not an action potential. It's a composite of all action potentials at a given time. So here the electrodes are placed at various points on the body to measure the voltage differences. So that's why there's these spikes here, because that's detecting the voltage differences. So during the P wave, that's during atrial depolarization. The QRS complex, that's during ventricular depolarization. And then there's the T wave that occurs during the ventricular repolarization. So you probably notice that there is no atrial repolarization. There's no spike for that. And that's because it occurs at the same time as ventricular depolarization. So that's why there's no spike here. Okay. Here you can see normal and abnormal ECG tracings. So this is a normal one where you have a P wave, QRS complex, and a T wave. But there's other situations where that could lead to abnormal readings or tracings. So like junctional rhythm, that's when the SA node is non-functional. So it's not producing that spontaneous impulse. And so this results in P waves being absent. Then there's second degree heart block. Here the AV node uh, doesn't conduct some of the SA node impulses. Because remember, the pulses begin at the SA node, the sinoatrial node, and then go to the atrioventricular node. But if some of those impulses fail to reach the AV node, it results in more P waves and QRS waves. Then there's the ventricular fibrillation, which is because of the electrical activity being disorganized and the action potentials are occurring randomly in the ventricles. So systole is the period of heart contraction, and diastole is the period of heart relaxation. In the cardiac cycle, blood will flow through the heart during one complete heartbeat. So atrial systole and diastole are going to be followed by ventricular systole and diastole. And then the cycle represents the series of blood pressure and blood volume changes. So these are the mechanical events that follow the electrical events that are seen on the ECG. The two sounds that you hear during a heartbeat are due to, um, well, the first one is due to the closing of the AV valves and at the beginning of ventricular systole. So again, systole meaning contraction. The second sound is due to the closing of the semilunar valves at the beginning of ventricular diastole. So ventricular relaxation. Heart murmurs are abnormal heart sounds that are heard when the blood hits obstructions. So this usually indicates valve problems. So for example, there's incompetent valves which fail to close completely, and that allows for a backflow of blood. And when that happens, it causes a swishing sound to occur as the blood is going backward from the ventricle into the atria. And then there's the stenotic valve, which this is a valve that fails to open completely, and that restricts the blood flow through the valve. So it causes a high-pitched sound um, as the blood is being forced through a narrower valve. Cardiac output is the amount of blood that's being pumped by each ventricle in 1 minute. And this is calculated by multiplying the heart rate times the stroke volume. And the stroke volume is the amount of blood or the volume of blood that's pumped by one ventricle with each beat. The three main factors that affect stroke volume are preload. So this is the degree to which the cardiac cells are stretched just before they contract. Then there's contractibility, which is the contractile strength at a given muscle length. And there's afterload, which is the pressure that the ventricles have to overcome to eject the blood. Heart rate can be regulated by the autonomic nervous system because the sympathetic nervous system can be activated by things like emotional or physical stressors. And also the parasympathetic nervous system counteracts the sympathetic nervous system. And there's the chemicals So things like hormones and ions like the calcium and potassium. Okay, so these are the main ones that regulate the heart rate. Okay, so I'm going to split this video into two. The first part is going to be the cardiovascular system related to the heart. The next one's going to be the cardiovascular system for the focusing on blood vessels.